All right, so this file is meant for a demonstration of the YASARA program that you will use to analyze protein structures. All right, so you'll get access to this YASARA program on either a server or one of the computers that we have here, but just as a reminder, this YASARA program is not meant to be copied, and so it needs to stay within the system. All right. So you'll have a screen like this when you open up the YASARA, and one thing that you can do that has been recommended is go to View, you can go to Color, and you can go to Background, and then you can change it to just white, and that'll change everything to look like that. And so the point being is that if you produce a figure or a image that you want to be able to produce or to use for a publication or a poster, you should start off with a white background so that there's no distortion, or it's easy to see, and you don't have to go back and recreate the image so that it works on in a PowerPoint or whatnot. All right, so you can start off with that. And then to get to the protein file that you're looking for, you'll go to Load PDB File, and you can find the file that you're looking for. All right, so I have a KRAS PDB file that I was able to access. Actually, I got this PDB file from a database that one of our colleagues has, and so you can request this and see if we can find the same PDB file for any other protein. We have a limited number of proteins that have been analyzed this way, but we could check to see if, so that might save some work if you need to have it. There are a few things that you can do to this image in terms of trying to orient yourself or changing the way it looks. You can go to this help and you can play the help movie, or you can go to about or show the user manual, and the user manual will open a web page which will give you a bunch of different links that you can get to. There's a lot of information here. And so if you have any questions, you could probably go here and find the answers for them. Otherwise, you can go to the help and play movie, and it'll demonstrate how you can manipulate the images that you have on the YASARA file. So for instance, you can rotate this, so that's a left click. You can zoom in or zoom out, and that's a right click while you're doing this. And then both clicks, both right and left click, will move from side to side. So that's just a beginner's aspect of how you can use the YASARA to look around at different things. All right. Notice over here you have this objects, and so if you click here, you can change different things that you might see by clicking the visibility. All right, so this one here is demonstrating the side chains. So if you click that, you just have the backbone. All right, so that gives you some kind of perspective. And so one thing that you can do, if you bring the cursor down towards the bottom, you have a list of all the amino acids in this protein, right? And so you can select one of these. So let's do valine. And so it'll zoom in, or it'll sort of have a little indicator where that valine is. So it's right here. And so you can do different things as you're going to this valine. So you can right click, and one thing that you can do is swap, and you can change this valine for any other amino acid. All right. You can also change the color of the residue, and you can change it to say red. And so now this valine is turned red. If you are particularly good at programming, you can double click the space bar and you have the code that's involved with all the different commands that you have. So if you want to incorporate your own code, then you can use it that way. And as a side note, a lot of what I'm talking about here is indicated in the condensed code slides PowerPoint that I have already sent you. All right, so we have a structure here, and there are some variants that have already been compiled for different proteins. So again, talk to us and we can let you know whether or not the variants for each protein have already been compiled in an Excel document. And so if I bring over the Excel file, you can, in this case, it is, I forget which one it was. This is TP53. And so this has a spreadsheet of all the different variants that they can do, and you can sort them. And then you can add, in one of the columns, you can add show res as indicated here. So equals, in quotes, show res and A2, which is that cell here. And so it will give you this result in the cell. And then you can drag it out. And so all these results can be brought down into this, this column here, right? And so then you can copy all of these and put them into YASARA in this quote, this part of the code structure. So you just copy and paste, and you'll end up with all of the residues in the protein turning a grayish color. Then you could go back to your spreadsheet and you can put in this code, which is equals, in quotes, color res space and A28. And so then you can do the same thing and you can change the color for a bunch of different amino acids. So in this case, we're changing it to red for the first 3, and then we're going to change the rest to yellow, right? And so again, you can copy and paste it into your the code area, and you end up with something that looks like this. And so in this case, the higher importance variants have been labeled most importantly with red and with varying degrees yellow in different spots, right? And so you can see that some of these residues are located in a specific three-dimensional area of the protein. So if you've done all this work, you can save. So you have an exact view. And so again, so you're going to go to File, Save as YASARA Scene, and so that will save the scene for you. And so you can save it wherever you want. Right, and so there's a way to get, so if you look at this figure here, it's sort of pixelated, right? So if you were to publish that, that would look a little bit poor. So if you want to save a high-res version of this, you could save as and ray trace high-res screenshot. And then you're going to run this POV ray and save ray traced image, and then you can press OK. And so that will give you this really blown up view. But then once you bring it down to the regular size, see how, you have pixelations here a little bit, but then once you make it smaller, then you're going to have a more high resolution image. Right? Say you want to rotate, have a rotating movie of your protein. You go up here. So see how you have this rotating symbol up here. And so that will rotate your image. So at the same time, you can save a movie of it while it's moving. So you could save as and MPEG movie, and then you could save it to the desktop or wherever. All right, so you could save it that way. All right, so you say you don't have a PDB file for the protein that you're analyzing. So you can go to UniProt or NCBI or any other bioinformatics that provides the FASTA format for different proteins. And so with UniProt, you can go to sequences down here and get a FASTA file. So you right click and you save that elsewhere. All right, and so you can go to options, choose experiment, homology modeling, and so you can find a FASTA file of your protein of interest, okay, and then you can select all there, and just save that there. In this field, this is the default is pretty much what you want for a high quality image or three-dimensional representation. If you want a quick analysis, you could just bring these numbers down to 1, and so it'll be fast, but it will may not be accurate. All right, so you can do that, and you'll start going through a whole lot of analysis. And so this I won't go through all this, and I won't let it run throughout, but you'll get to a point where you'll see the computer producing a bunch of images, and it's rotating and it's changing the structure. And so it's basically fitting the FASTA sequence of the amino acids into structures based off of homology of other proteins. When you're done with the structure, you need to save this and save as a PDB file. And you can find the location of wherever it's supposed to be saved, or you select the one that you want to save, and you need to select the folder that you need to save it, and it'll produce the file that you'll use later on. So if you were to go through that process, you get a folder with a whole bunch of data in it. You'll have your PDB file there, plus a bunch of modeling information. If you go to this, your model, the HTML file, you'll have this data here, which will give you a bunch of information about the protein structure. As probably mentioned before, there's I-TASSER, which is a program that does something similar. It will produce a PDB file based off of another method of producing three-dimensional structure. And so you can maybe use that for confirmation, or if the YASARA program didn't work, you can produce a PDB file that way. All right, so you may have obtained a PDB file from one of the databases that we have here, but sometimes the PDB file is not perfect in terms of the arrangement of the atoms as well as the placement of the different atoms. And so if you look at this image here from a PDB file, if you look closely, you can look at the residues, and you can see that the hydrogens are not present in this image. And so that's one of the things that needs to be corrected with the PDB file before you start analyzing things. So first thing that we would do with this PDB file is go to force field here, and for this specific type of program that we have, we'll use the YASARA force field here and press OK. Right, and then we will go to options and macro and movie, and then play macro, and then we can run this em run clean mcr file. All right. So see, now that they've added the hydrogens, and now we're having waters, and we're gonna bring the molecule to its lowest energetic state. Okay, this process takes a little, a few minutes, but now you can see that the process is finished. The molecules have stopped moving on the display here, and you have this message at the bottom indicating that you are completed. All right, so now you can go and save your PDB file. And I just save the image itself. I don't save the waters here. All right, and then I could save it whatever. So make sure that you go to the appropriate folder and you change the name of whatever file you have into whatever you want. So you can press OK. So at that point you will have a new PDB file. All right, so you'll be given a MD run code MCR file, which is basically a script that has a whole bunch of commands in it that adds extra data to your analysis. All right, so if you go to options and macro movie, and you can set your target. So I'll set the same protein as the target, and then we go again to options, macro movie, and then play macro. All right, and so then you could find that MD run code MCR. All right, and so I have this right here, and you press OK. All right, and then it'll start analyzing this protein. All right, and so this is a process that will take quite some time. It may take days or it may take weeks to analyze a protein depending on its structure and complexity. All right, so you would run this and let it go, and it'll run different steps of this code so that you eventually, so don't try to move things around because that changes how things work here. If I were to show the residues for this protein, and I'm running this modeling program, then you should be able to see the calculations that are being made by looking at the residues, and you can see they're jumbling around. So basically it's going through different configurations of these atoms at considering every atom, and also what I'm not showing here is all the water that is surrounding this structure. So it has to keep into account the waters that are dissolving this protein. And so this does this for 20 nanoseconds of time, and then it produces data. But yeah, as mentioned before, this would take days or weeks. So depending on the size and the structure and the complexity, it'll determine how long this processing will occur, and also how many cores you have working on this also. All right, so I'll just stop this just to we don't want to go through the entire thing. So what you will end up having is this report that will give you all the different simulation data. You'll get this HTML file that will give you all some of the data regarding all the information that was produced during the simulation. You can get files like this, or you'll have an image like this which shows the interaction of different amino acids with each other. So on the X and Y axis, you have all the amino acids for that particular protein listed out. And so some amino acids interact while some of the others do not, right? And so you can imagine what you can do with this particular diagram here, where if you introduce a substitution for a different amino acid, you might see a change in this diagram, which indicates a change in the structure.